Good morning. This is Lecture 7 for Part 1. Last lecture we talked about ligation mixture and we said that in our ligation mixture we might have the original plasmid. Also we'll have the unligated fragments like unligated plasmid and unligated gene of interest. Unligated DNA contaminant is possible as well. Also, we can have recombinant DNA, either with the right insert or with the wrong insert. Here, number 1, you have the original plasmid, which contains the tetracycline-resistant gene and the neomycin-resistant gene. This original plasmid, we cut it by restriction enzyme. We digest it by a restriction enzyme, which opens the neomycin gene, neomycin resistance gene. Why is that? Because the neomycin resistant gene will have the MCS region, which is the multiple cloning site. So the digested plasmid will be cut at the neomycin resistant gene, just at this place, while the tetracycline resistant gene will be intact. If we add the ligase and the gene of interest, which is here the gene of insulin, is inserted inside the plasmid, we'll have the recombinant DNA with the correct insert, which is number 3, the recombinant DNA with containing the gene of insulin. Sometimes number 2, which is the digested plasmid, will be self-ligated, which means it will go back to number 1. It will go back as the original state. It will be exactly as the original plasmid. So now imagine that the original plasmid or the self-ligated plasmid was inserted inside the bacteria cell. The bacteria cell will now gain the resistance feature for the neomycin gene and for tetracycline. It will be resistant to tetracycline and it will be resistant to neomycin. And of course, it will not have the ability to produce insulin because the insulin gene is not inserted inside the original plasmid or the self-ligated. Now imagine that the digested plasmid, which is number 2, or the unligated plasmid and the unligated gene of insulin or any other unligated DNA fragment was taken up by a bacteria cell. These unligated fragments are most likely to be degraded by the enzymes that are in the cytoplasm of the bacteria. So this bacteria will not gain any resistance feature. It will not be resistant to tetracycline and it will not be resistant to neomycin. It will not have the time to express these genes. Why? Because the enzymes, the degradative enzymes inside the bacteria will take or will degrade these DNA fragments. Now imagine that the recombinant DNA is inserted inside the bacteria, or the bacteria took this recombinant DNA inside it. The recombinant DNA, as you can see, it has an intact tetracycline resistant gene, which means this gene is active and this bacteria will gain the feature of resistance against the tetracycline. So if we put it in a media that contains tetracycline, it will definitely live. On the other hand, the recombinant DNA has the neomycin resistant gene, but it's inactive. Why it's inactive? Because there is a gene of insulin inside this neomycin resistance gene, which means this neomycin resistant gene is disturbed. It's not active. So if we put the bacteria in a media that contains neomycin, it will be sensitive to neomycin and it will die in this media. Can this bacteria express insulin? Yes, definitely it can. Why? Because it has the gene of insulin. So the difference here between the number 1 and number 3, which is the cell that contains the original or the self-ligated versus the bacteria cell that has the recombinant DNA, number 1 will be resistant to neomycin and it cannot express insulin, while number 3, it will be sensitive to neomycin, but it can express insulin. That's why in lecture 4 we said that the antibiotic resistance gene are selectable markers, because here, as you can see, from the antibiotic resistance gene we can differentiate between the cells or the transformed cells that take up the recombinant DNA or the transformed cells which take up the original plasmid. We know that if the cells took the recombinant DNA, they will be sensitive to neomycin, while if they took the original plasmid or the self-ligated ones, it will be resistant to neomycin. This method is called selection of desired cells, or the screening. We want to screen which cells have the recombinant DNA. You might ask me what will happen next if our transformed cell, which has the recombinant DNA, is dead. What's the point, right? So usually we culture a large number of bacteria, and from this large number we test a small number, or we take a sample from our batch. From this sample we know that our batch has the recombinant DNA or the original plasmid. Also, you might ask me why we have two selectable markers, why we have two antibiotic resistance gene. The first one, the neomycin resistant gene, is used to differentiate between the transformed cells, which of them have the original plasmid and which of them have the recombinant DNA, because we know that the neomycin resistant gene will have the insert if the ligation went well. The other antibiotic resistant gene, the tetracycline resistant gene, is used to get rid of the non-transformed cells. We'll have a large number of cells that did not take any DNA inside it, that did not take the none of the original plasmid or the recombinant DNA. So in order to get rid of all the normal cells or the cells that did not take up any DNA, we'll put all the cells in a tetracycline media. If the cells are dead, these cells will be the normal cells, while if the cells live, this means that these are the transformed cells, and the next step will be to differentiate which of these transformed cells have the original plasmid and which of them have the recombinant DNA. So here you have a question regarding screening or selection of desired cells. We have cell number 1, which is a normal E. coli cell. Normal means it did not take up any DNA. It doesn't have any plasmid. It doesn't have any recombinant DNA. It's a cell without any foreign DNA. Cell number 2 is a transformed E. coli cell with self-ligated. And cell number 3 is a transformed E. coli cell with the correct recombinant DNA. Here they said the gene to be cloned is insulin gene, and the vector used is shown in the figure. So let's have a look at the plasmid figure. Our plasmid has 5 areas or 5 regions. The first one is a promoter, which is used to drive gene expression. The second one is the tetracycline resistant gene, which includes the MCS, which means the multiple cloning site, and this site is where the restriction enzymes cut the plasmid. Number 3 is the ampicillin resistant gene, and number 4 is the ORI, which is the origin of replication. So the first cell, cell number 1, which is the normal E. coli cell, we said that it does not have any plasmid, it does not have any recombinant DNA, so it did not gain any resistance feature. For that reason, cell number 1 will be sensitive to ampicillin and it will be sensitive to tetracycline. So if we put them in a media that contains either an ampicillin or a tetracycline, cells number 1 will definitely die. The second cell is a transformed E. coli cell with self-ligated. Self-ligated plasmid means we'll have the original plasmid, although it was cut by the restricted enzyme, but it was ligated on itself and gained all the original shape of it. So it will have an intact tetracycline resistant gene and an intact ampicillin resistant gene. They will not be disturbed. These antibiotic resistant genes are not disturbed by any insert. That's why the E. coli cell which has the self-ligated plasmid will gain the resistance feature. It will be resistant to ampicillin and it will be resistant to tetracycline. So in a media that contains both antibiotics, it will definitely live. The third cell is a transformed E. coli cell with correct recombinant DNA, which means our DNA will have an intact ampicillin resistant gene, but it will have a disturbed tetracycline resistant gene. The tetracycline resistant gene will be inactive. Why is that? Because the gene of insulin is inserted in number 5. It's inserted in the MCS and disturbed the tetracycline resistant gene. So the recombinant DNA lost the resistance feature, and our E. coli cell will be sensitive to tetracycline, although it will be resistant or stayed resistant to ampicillin. So if we put the cell in a media that contains ampicillin, this E. coli cell will live, while it will die in a media that contains the tetracycline. If we put both antibiotics, it will definitely die. Again, here this figure represents the gene cloning. We isolated the plasmid DNA and the human DNA. The plasmid DNA here contains ampicillin resistance gene, and also it has a LAC-Z gene, lactose breakdown, or a gene that's responsible for lactose breakdown. This is another selectable marker that can be used in order to differentiate the cells according to color. Okay, I did not mention that before, but just to know, this is one of the ways to screen cells by not killing them, but by differentiating between them by colors. So after that, we cut both DNA samples with the same restriction enzyme in order to have sticky ends, preferably. After that, we joined the DNAs by ligation, or by ligase enzyme, which builds phosphodiester bonds between the gene of interest and our plasmid. This will produce a recombinant, or will generate a recombinant DNA. The final step is introducing this recombinant DNA into bacterial cells. After that, of course, we'll have a series of purification steps that we'll talk about in part 2. So how to select our expression host? The expression host is the host cell that is used to express the gene of interest. For example, we said that we can use E. coli in order to express the gene of insulin, which means we want to produce insulin inside the E. coli. We always prefer the E. coli or the bacterial cells over other host cells. Why is that? Because it's easy to manipulate, it's fast to reproduce and divide, etc., etc. However, sometimes we cannot use bacterial cell as an expression host cell because it cannot perform post-translation modification. Post-translation modifications are processes that are performed after translation process. Some of them, for example, glycosylation. Some proteins need to be glycosylated after translation, which means adding some sugars to the structure for its biological function or stability. Other proteins need disulfide bonds, others need phosphorylation. All these processes are performed after translation. And this cannot be done inside bacterial cell. Usually we need a more complicated cell, such as mammalian cells. We have many expression host cells. Like for example, proteins can be produced in E. coli, yeast, plants, mammalian cells, or even transgenic animals. We'll talk about each expression system in part 2. The most famous, or one of the most famous cells or mammalian cells used in gene cloning is the Chinese hamster ovary cells, the CHO cells. At first, scientists were not encouraged to use mammalian cells. Why is that? Because they need a long time to divide, they are difficult to culture, in addition to other problems like needing expensive media and many other problems. We'll talk about them in part 2. On average, here they said mammalian cells divide only once every 24 hours, while cell division E. coli takes around 30 minutes. Some bacterial cells, for your information, they take only 17 minutes. And also here they said that the yeast take around 1 hour every cell division, for a cell division, which is relatively fast. That's why we prefer E. coli, and we are not very encouraged to use mammalian cells. Also, mammalian cells need expensive growth media, such as bovine fetal serum, which is serum which was taken from the cow fetus. Why we use the serum? Because it's filled with the growth factors and vitamins and all the factors needed for the growth of mammalian cells. The problem with bovine serum is that it can be easily contaminated with microorganisms such as bacteria, fungus, viruses. And the worst case scenario, it may contain prions. Prions, if you remember, they are infectious proteins and they can cause mad cow disease. Mad cow's disease, here they said it has another name, bovine or transmissible spongiform encephalopathy, BSE or TSE. So we have to be careful if we want to use a serum. We need to know from where to buy it and how to deal with it. So now biotechnology companies, they try not to use bovine serum for all the issues that it has. They try to build their own or create their own defined media. They add all the growth factors needed, all the vitamins and any other components that we need for mammalian cell growth. So as we said before, we can buy the serum from certain countries with the lowest possible TSE risk or the lowest possible mad cow disease risk, such as the USA, Australia, and New Zealand. This table represents the pros and cons of different expression hosts. The first expression hosts are the prokaryotes. And the most famous example for prokaryotes are the E. coli. The pros or the positivity of using E. coli, they are easy to manipulate. They can grow rapidly. They can divide fast. Overnight we can have a huge number of E. coli cells. Also, we can have large-scale fermentation. Overnight we can have large amount or big number of bacterial cell, which express all of them express the gene and produce the desired protein. Also, we need simple media to grow these cells. We need simple salts, simple food with lowest risk of contamination. So maybe the only risk that we can have here is the human handling, actually. So it's very simple, it's easy to use, easy to manipulate. Also, we can have high yield of the protein. We can use large amount of protein from this small cell. However, the negativity or the cons of using such cells are sometimes we can have misfolded proteins, which means the proteins may not fold correctly. This happens because E. coli cell, for example, is very small in size, and it cannot perform post-translation modification, which are necessary for protein folding. Like, for example, formation of disulfide bonds are very important for protein folding. That's why it may aggregate. So in bacteria, usually we can have inclusion bodies, which are aggregates of the protein. The protein will aggregate on themselves. Also, it says here we'll have almost no post-translation modification. Why almost? Because some bacteria, not the E. coli, but some bacteria may perform simple glycosylation. Okay, so it's not always no translation modification, but some bacteria may perform glycosylation, but very simple glycosylation. The second expression system is the yeast, like for example Saccharomyces cerevisiae. This is also used, by the way, to produce insulin. I think the company Novartis uses yeast to produce the therapeutic insulin, the recombinant insulin. The good thing about the yeast, it's also grow fast, relatively rapid, or relatively fast. We can have larger scale fermentation, yani we can have a large number of yeast cells. They all express the gene of interest. Also, the good thing about the yeast that it can perform some post-translation modification, which is a good thing. However, the cons here, they said that the post-translation modification may differ from humans, especially the glycosylation. So we'll not get a perfect glycosylation. That's why sometimes we cannot use the yeast, and we have to use the mammalian cells if we have very complicated glycosylation form. The last expression system is the mammalian cell. The mammalian cells, such as Chinese hamster ovary cells or human embryonic kidney cells, they may grow in suspension. We can perform all required post-translation modification, although they will not be identical to human post-translation modification or the endogenous proteins, or the glycosylation for the endogenous proteins. However, they are good enough as therapeutic proteins. Okay, so this is the perfect cells or the perfect scenario for glycosylation. However, we have many cons in here, like for example, slow growth. As we said, mammalian cells, they divide once every 24 hours, so we'll need more than 2 weeks in order to get a good amount of mammalian cells, and of course even more if we need to produce a large amount of our drug. Also, the mammalian cells need expensive media, and especially the serum. If we use the serum, it's expensive and it has the highest possible risk of having prions. contamination. Also, it's difficult to scale up. We'll talk about scale up in part 2. And here they said also dependence of serum. We already said that. The main reason why mammalian cells are used as an expression host cell is that in these cells, post-translation modification PTM of the synthesized proteins resembles most closely the human situation. What does this mean? Post-translation modification is the modification performed after translation, as we said before. We have many post-translation modifications, but we need to know that we always produce the therapeutic protein according to the endogenous protein. If the endogenous protein is post-translated, we have to produce a post-translated therapeutic protein, which means, for example, our insulin is not glycosylated. The endogenous insulin, the insulin that we produce inside our body, the original human insulin, is not glycosylated, but it has disulfide bonds. So when we produce a therapeutic insulin, it has to contain disulfide bonds, but it should not be glycosylated. So the therapeutic insulin should resemble the endogenous insulin, or to some degree of similarity. On the other hand, for example, we have erythropoietin, which is a glycoprotein. This glycoprotein means that this protein has glycosylation or sugars in the structure, and they are necessary for the function, for the stability, for many features of this protein. It will be only functional if it's glycosylated. That's why when we produce the recombinant erythropoietin, we build it as a glycosylated form. It's not functional if it's not glycosylated. So the therapeutic protein should resemble the endogenous protein, or to a high degree of similarity. Of course, we cannot perform a high degree of glycosylation, or we cannot control glycosylation, but it's acceptable to certain level. An important PTM is the formation of disulfide bond between two cysteine moieties. It's not performed between just the two cysteine. It can be performed between two methionine, between a cysteine and a methionine. Cysteine and methionine are two amino acids that have the sulfur atom. That's why they can perform they can be involved in the formation of disulfide bonds. And disulfide bonds are crucial for the stability of the tertiary structure. Here they said that E. coli is not able to make disulfide bonds in a protein. And already for this reason, E. coli is not very suitable for producing most of the marketed therapeutic proteins. You may wonder how can we use E. coli to produce insulin, yet insulin has 2 or 3 disulfide bonds in the structure, right? Insulin, what happens here, the structure of insulin has 2 chains, chain A and chain B. Usually, the company produces chain A, or the polypeptide chain A, in a cell, and chain B in a different cell, in a different bacterial cell. After these 2 polypeptides, or after these 2 chains are produced in 2 different cells, they isolate them, then they put them in reducing condition in order to perform the disulfide bonds. So the disulfide bonds are not formed inside E. coli. They are formed after the production or after the expression of these two chains inside the bacteria cell. Another important post-translation modification, as we said before, is the glycosylation. And as they say here, around 70% of all marketed therapeutic proteins, including monoclonal antibodies, which we'll talk about them later on, are glycosylated. Notice that 70% of all marketed therapeutic proteins cannot be produced inside bacteria because they need glycosylation. Okay. If we want to define glycosylation, it's the covalent attachment of oligosaccharides to either asparagine end-linked or serine threonine O-linked. This means that glycosylation means adding sugars, or here oligosaccharides. How do we add oligosaccharides? We add them by covalent bonds to one of three amino acids: asparagine, serine, or threonine. Asparagine, if we want to add sugar to asparagine, we add it to the N atom in the R chain of the asparagine. You'll understand this in the next slide. However, if we want to add oligosaccharide on serine or threonine, we'll add these sugars on the O atom, on the oxygen atom in the R chain of our amino acid. Why glycosylation is important? It says here they affect many of its pharmacological properties, including stability, solubility, bioavailability, in vivo activity, even pharmacokinetics and immunogenicity. So we cannot ignore this element of the structure. We have to have the glycosylation if the endogenous protein is glycosylated. This figure shows the three or two amino acids. The asparagine, it links with the oligosaccharide, with the sugar, by the N atom in the R chain. Asparagine is an amino acid. Okay, it has the basic structure of C alpha linked with the amino group from one side and a carboxy group from the other side and one hydrogen from the third side, and the fourth side has the R chain. So the R chain has an N atom. The nitrogen atom links with the sugar. On the other side, we have also the serine. Serine is also another amino acid. It also has the C alpha, which is linked to the amino group from above, the carboxy group from below, and the hydrogen group from right side. And from the left side, it has the R chain or the side chain. The side chain has an oxygen, and from this oxygen, the sugar will be linked. So this means the O linkage or the O link glycosylation, or the asparagine when we say the N glycosylation, which means the glyco or oligosaccharides added to the N atom of the R chain of the asparagine. They also say here that the glycosylation differs between species, between different cell types within a species, not only between species, and even between different cell types within a species. This means if we took cells from ovary of a monkey, or we took cells from the kidney of a monkey, each cell type will give us different glycosylation. And not even that only, they say here that even between batches of in cell culture produced therapeutic proteins, even if we have the batches from the same original, they may give us different glycosylation. So glycosylation cannot be controlled. There are no rules that says in this pathway we'll have this glycosylation, or these sugars will be added, or this number of sugars will be added. We cannot control them like we control transcription or translation processes. That's why glycosylation is always a complicated process to perform. This figure shows folded structure for two proteins. The N letter represents the asparagine and the ST represents serine or threonine. As we said, the asparagine will be linked to oligosaccharides by the N atom in the R chain, while the serine or threonine will be covalently linked to oligosaccharide by an oxygen in their R chain. And try to have a look at the number of sugars or the type of sugars that were added in each amino acid. Even in the same asparagine, in two different asparagines, you'll have different sugars, you'll have different numbers of sugars. So there are no rules at all of how to add oligosaccharides or how to perform glycosylation on these amino acids. So this is the end of Lecture 7. If you have any questions, you can contact me by email. Good luck!